Hello guys, welcome to my channel drStingRay, and hope you are all doing well. So let's talk about today's lesson. Today we'll talk about the nervous system. Let's proceed forward. So first of all, we need to know, why do we need a nervous system in our body? So what are the functions of nervous system? Functions of nervous system. First thing is it helps us to keeps us informed about what is happening in the outside world. Okay, so it keeps us informed about the outside world. Secondly, it helps us to think. It gives the ability to think. Thirdly, it helps us, or rather it coordinates the various voluntary movements of our body. Say I want to a ball is there on the ground, I want to pick it up. I need muscular activity, that is controlled by the same nervous system, that is voluntary controls. It also controls the different involuntary functions of our body. Say when we eat the food passes down the alimentary canal using the process of peristalsis. That process is also controlled by the nervous system. Say there are various secretions from various glands, that is also controlled by the nervous system. Okay, so we have the involuntary controls. So these are the basic functions of the nervous system. Now you must have heard that cell is the structural and functional unit of life. So when many cells are taken together, okay, are bunched together, they form a tissue, an organ system, and ultimately an organism, right? We have learned this in junior classes. So it is the structural and functional unit of life. So in the similar way, the nervous system also has a unit. Okay, the unit of nervous system. What does it mean? That when the unit is repeated, okay, continuously, it forms the entire system. So what is it? It is called a neuron or nerve cell. Okay, so let's talk about the neuron. Now, a neuron presents with three parts. What are the three parts? The first one being the cyton, that is also known as the cell body, or it has another name. What is it? Perikaryon. Perikaryon. Second we have the dendron, which ultimately gives rise to dendrites. So we can also write here dendrites. Okay, now the third one being the axon. This is the longest part of a neuron. Now we'll talk about it individually. Okay, so first let's talk about what is a cyton. Okay, cyton is the cell body. We will show it using a diagram, right? So let's draw a diagram of a neuron, basic diagram of a neuron. Okay, What I am drawing now, we'll talk about it later, right? What is it? You have a nice nucleus. So this is the picture of a neuron. Now we are concerned here with this part. This is known as the cell body or the cyton. This is known as the cell body or the cyton. Now what is present in the cell body? There is a fine nucleus, as so does all the cells have. Similar way, the cell body here also has a nice nucleus. Okay. Next, what we have, it is important to know, we have small granular structures in the entire cell body. What are these? These are proteinous granular structures called, these are called Nissls granules. These are derived from the ribosomes. Next, what is, and the cell will also contain all other cell organelles which a normal cell contains. However, what is important to know is a normal animal cell also contains what we call as centrosome, right? What it helps in? It helps in cell division. However, in nerve cell or a neuron, there is no centrosome, right? There is no centrosome. As a result, it is not capable of dividing. So it's an important characteristic of a neuron that a neuron can never ever divide, right? So that's all about the cyton. Okay. Now let's talk about the dendron. Which part of the cell is dendron? See here, these parts which are projecting from the cell body, they are known as dendrons. Okay, these parts are the dendrons, and when they give finer branches, they are called dendrites. Correct? It's important to know here also that these dendrons also have Nissls granules. Dendrons also have Nissls granules. Next, that's enough for the dendrons or the dendrites. We'll talk about their functions later on, okay, because they are interrelated. Now let's talk about the last one, that is the axon. You see this long process, that is the axon. Actually the projections of the cell body, you know, these projections have developed into dendron and one of them have developed into axon. So it is also a projection similar to the dendron, but it has developed longer, so it is known as axon. And there is no limitation that a single neuron will have only one axon. It can have multiple axons. Based on that, even we have classification of neurons, which is of course the out of the scope of syllabus for now, but it's important to know that the cell may have more than one axon. So now, so it's the longest part. Let's first write longest part. Okay, now this axon is a fine structure. Okay, surrounding it, what we have is small cells. Okay, small cells. These cells will also have a nucleus. What are these cells called? These cells are called as Schwann cells, named after the scientist Schwann, SCHWANN, Schwann cells. Okay, now these cells may secrete a fatty substance called myelin. You know, fatty substance called myelin. They may or may not secrete. In that way also we have another classification of neuron. Myelinated if myelin is present, and non-myelinated if myelin sheath is absent. We call it myelin sheath. It acts as a cover, so we call it a sheath. Myelin sheath. What is it? It is a fatty substance. Okay. Now this entire thing in this axon part is again covered by a membranous structure. Okay. What is this membranous structure called? This membranous structure is known as Neuro Lemma. Neuro Lemma. Now you must have noticed, let me just notice this part, okay, the junction between two Schwann cells. I'll just redraw it and just magnify it. Okay, let's see how it looks. I'll just magnify it. So this is the axon. Okay, these are the two adjacent Schwann cells. Let me make me bigger. These are the two adjacent Schwann cells. Now the Neuro Lemma comes, and at this junction it makes contact with the axon fiber. Okay, it makes contact with the axon fiber. So this junction, this has a special name. Okay, we call these junctions as nodes, so we call it as node of Ranvier. Node of Ranvier, of course, after its discoverer. Okay, what is the usefulness of this node? No, it helps in saltatory conduction of nerve impulse, which is very fast. You know, you will learn about it in higher classes. Saltatory conduction means the impulse will jump from one junction to other. See, the axon may be much longer. Okay, it may be up to 1 meter, from millimeters to meters. Okay, so the nerve impulse jumps from one junction to another. Okay, this type of transmission of nerve impulse is called saltatory conduction. We will come to know about this in your higher classes, right? So this saltatory conduction is known as saltatory conduction. This saltatory conduction is mainly caused by this junction, right? This junction. So that's all about the axon part, and that's all about the parts of a neuron, right? Now let's talk about how a nerve impulse is generated and how is it transmitted. Okay, say we saw the axon as a simple thin fiber. Okay, we are redrawing it very large. Okay, so this is the entire axon fiber. This is the entire axon fiber. Okay, now you see, this is the entire nerve fiber. So normally you see there are some channels out in this membrane. Okay, there are some channels in this membrane. These channels, what are they known as? They are known as, I am writing it here. Okay, these channels are known as sodium potassium ATPase or ATPase pump. What are these known as? Sodium potassium ATPase pump. What is the function of this? Now this pumps 3 sodium ions. Okay, 3 sodium ions from inside of the axon to outside, and simultaneously together it acts together. Okay, it just pumps 2 potassium ions inside of the cell. So 3 sodium ions going out, 2 potassium ions coming in. So you may notice that there is a relative loss of positive charge inside. How? Let's take an example, a fine example. See, sodium and potassium both have one positive charge, right? Both are equivalent in that. So let's draw 5 positive signs in, okay, 5 positive charges inside, 5 positive charges outside. Okay, now what I said, from outside to inside 2 potassium ions will come inside. Okay, right? So let's consider these 2 as potassium ions. They came inside, so they are erased from here, and I draw it here with the red, and 3 sodium ions will move outside. So say these are the 3 sodium ions, I take them outside. Okay, this is happening through the sodium potassium ATPase pump. Now so let's erase these 3 and redraw it here using blue. 3, 1, 2, 3. Now let's count the total positive charge of outside and inside. See, inside we have 1, 2, 3, 4, and here we have 1, 2, 3, 4, 5, 6. So initially we had 5, 5. Now we have 6 and 4. So there is a net gain of positive charge outside, and there is a net gain of net loss of positive charge inside. So what we call is, for our own convenience, what we call is that the outer part of the membrane is positively charged and the inner part of the membrane is negatively charged. Mind you, this inner part of the membrane is not actually negatively charged, but it has less positive charge than outside. So to make it easier for our own concern, we say that it has negative charge. By negative charge, we mean that relative to the outside environment, it has negative charge. Okay, say if it is having 4 positive charge, the outside will have 6 positive charge. So we are calling this as negative and outside part as positive. Okay, so let me redraw it once again and explain you the generation and transmission of nerve impulse. This is the axon. We have what? We have learned that initially there is positive charge outside. This is the normal scenario, and we have negative charge inside. Now say I put a stimulus. What is a stimulus now? Okay, what is a stimulus? A stimulus is something of the external environment that when we apply on the neuron, it generates an action potential. Now what is action potential? Action potential is a type of disturbance created here, okay, which will ultimately give rise to a nerve impulse, right? This nerve impulse is further transmitted and taken to the brain or spinal cord, wherever it is destined to be taken. Okay, so how is it generated? Now say I touch here, I give it a stimulus. What happens here is, here are some more channels, some more sodium channels. Okay, these are single sodium channels. These are what? Sodium channels. On providing stimulus, okay, these channels are opened up or activated. What this does, now we have sodium, excess of sodium outside and excess of potassium inside. So there is a sodium deficiency inside, right, due to the action of sodium potassium ATPase pump we have seen previously. So this excess sodium, what it does is, this excess sodium, this starts coming in through these channels, right? So there is an influx, we call it, the technical term for this is influx of sodium. So there is sodium inside. Now what happens? There was positive charge outside, now positive ions are coming inside. So what happens? This charge, this initial charge, that there is positive charge outside, this negative charge outside, this is hampered. Ultimately what is created? Ultimately what is created? Ultimately due to the influx of sodium ions which is positively charged, okay, there initially here it was positive charge, okay, here I applied the stimulus, sodium entered, that is the positive charge entered, and now the polarity of this part of the membrane gets changed. How it gets changed? Now excess positive charge now comes inside and make the outside part negative and the inside part positive, right? There is a reversion of polarity, right? It is not the normal condition. Here we have applied action potential, okay. Now this process is called depolarization of the nerve impulse. Previously what the condition of the nerve it was, we call it as a polarized nerve. Now when we apply the stimulus, this polarity is reversed here. This is known as depolarization, okay, depolarization. Now here this is all about the generation of the nerve impulse. Now how is it transmitted? See, you have learned about current electricity in physics, right? So current flows from positive to negative. Similarly here also these positive charges, these three positive charges will flow here to here, okay, and there are three negative charges here. So what they will do, these three next positive charges, okay, the next three positive charges will flow from here to here. What will the scenario? These three positive charges are shifted here and these three negative charges are shifted here. Why? The positive charges are coming this side, okay. What will it look like? Let me draw this part of the membrane just here, okay. I'll draw it. What will it look like? No, from here I am drawing. Here I have again three positive charges as before, and here I have again three negative charges as before. What is changed is now here there were positive charges which shifted here, so we have the new positive charges here, and we have the new negative charges here. So this depolarization, this wave of depolarization is transferred from here to here, okay. This happens very fast, very fast. Similarly, now let's finalize it. So what happens? Now what we have again three positive charges here and three negative charges here as usual as before, right? What has happened is now we have the negative charge which has traveled towards this side, and we have a positive charge which has traveled here. So this is the wave of depolarization which will get transferred from here. It was generated here, but it got transferred to the next part, okay. Similar way it will get transferred or transmitted throughout the nerve, okay, and it will move in a desired direction. How will it get transmitted? Now this is how it will get transmitted, okay. Now this is all about how it gets transmitted. Now I drew a picture of cell body, the neuron. Let me redraw it somehow. This was the axon, okay. These are the dendrons, dendrites, cell body. What happens is the nerve impulse travels from this direction to this direction, right? Travels from this direction to this direction. What happens is, see, a nerve impulse is generated in this neuron and it gets transmitted here. Now at the end these are bulbs, okay. These bulbs are called axon terminals, right? These bulbs are called axon terminals, okay. Now here maybe that another neuron, okay, another neuron, say it is the dendrite, here is the cell body, okay. Another neuron is connected here, or another neuron is connected here, okay, which is also connected to any other neuron. Maybe this may happen. So what will happen in this case? The nerve impulse will get transmitted from this neuron to the next level of neurons. Or it may happen that these axon terminals may be innervating, or these axon terminals may be supplying the muscles. So here may be the muscle. Okay, ultimately it is giving impulse to the muscle. Say I have to contract my muscle, I have to just do this kind of movement. Okay, my nerve impulses are getting transferred and it is innervating the muscle. So the nerve impulse is telling my muscle that you contract. Okay, this is the function of this neuron, end neuron actually. Okay, this neuron has electrical impulses have already been transferred from other neurons to this neuron, and it is finally transferring it to the muscle, and the muscle is acting accordingly. Right, now this is what is all about the transmission of the neuron, or the the transmission of the nerve impulse. Okay. Now the nerve impulse got transmitted. Okay, but previously we had a wave of depolarization here, which is transferred to the next part, which is again transferred to the next part. Again it is a continuous process till it reaches the end of the neuron. Okay, now the part which was previously depolarized here, now it again got normalized. What is the importance of this? Until and unless it gets normalized, it is not ready for carrying another impulse. Okay, so it has to get normalized. What I called this process of getting hampered, the polarity getting reversed, I called it depolarization. So reversing it again, we get the normal polarity. So what should we call it? We call the previous one depolarization, so we will call this normalization of the polarity as repolarization. This is what is called as repolarization. Okay, now as I told, the nerve impulse will either get transmitted from one neuron axon ending to the other neuron, other level of neurons, or finally to the muscle. Now how does the impulse get transferred from, as far as it is inside the axon, it is traveling the way I showed you. Okay, it is getting transmitted as the way I showed you, but when it has to be transmitted from one neuron to another neuron at the level of the axon terminal, how it gets transmitted? Here comes the concept of a synapse. Here comes the concept of a synapse. Now you must have heard synapse and synaptic clefts. Mind you, these two are little different, so we have to know the difference between these two. So as I had drawn a neuron. Okay, this was the axon, here was the axon endings, axon terminals, here we had another new bunch of neurons. So we have only one neuron now for now. This is another neuron. I am drawing this part, the connection between the axon terminal of this neuron and the dendrites. Okay, so what is the function of dendrites then? The dendrites receives the nerve impulse from its higher level of neurons and it transmits it to the cell body. Okay, now this cell body transmits the nerve impulse further to the axon, and this axon further transmits it to the axon terminals. Now from the axon terminals it may go to another level of neurons, or it may go directly to the effector or the muscle or the gland, wherever it is destined to go. So I will draw this part when the nerve impulse travels from one neuron to another at this part. Okay, the axon terminal and the dendrite which is receiving the impulse from the axon terminal. I will just magnify this part, how this part looks like. It's a representational diagram. Okay, this is the axon terminal. All right, this is the axon terminal, and let's say this is the dendrite. The ending of the dendrite which is receiving the nerve impulse. Okay, how does all those things happen? See, nerve impulse is traveling from the axon, okay, this way. So nerve impulse is coming like this. it has come here. What is present here is there are numerous calcium channels present here. Okay, when a nerve impulse reaches in this area, these calcium channels are voltage gated, means they will be activated by an electrical or a nerve impulse. So when these nerve impulse reach here, these channels get activated, and there are numerous calcium ions, actually different ions present in the outside environment. So these calcium ions will leak, or rather be pumped. They will come inside. Why? Because there is lower concentration of calcium inside and higher concentration of calcium outside. So it will move from a region of higher concentration to a region of lower concentration. Okay, so we have calcium ions inside. Now we also have some vesicular structures here from beforehand. Okay, these vesicular structures contain chemicals which are called neurotransmitters. Okay, these are neurotransmitters. Now these calcium which have influxed here, these force these vesicles to move towards the end of the axon terminal. Okay, they will move towards the end of the axon terminal, and finally what will they do? They will fuse with the membrane of the axon terminal. Okay, I'm drawing some less of them just to show you. Okay, and then it contains neurotransmitters. Okay, finally it will fuse, the calcium will make them fuse with the axon terminal membrane, and finally after that this membrane will disintegrate, and this neurotransmitter which was present inside the vesicle will be thrown out, okay, into this space. This space is called as the synaptic cleft. This space between these two structures. And when we take these two structures, means the membrane of this axon terminal and the membrane of the dendrite as well as the synaptic cleft, we call these three structures together as the synapse. Okay, it is the functional gap between two neurons. Okay, so these neurotransmitters are now present here. Okay, again here are special channels. What are these channels? These are again sodium channels. These are activated by what? These are activated by these neurotransmitters. These will come and activate these sodium channels, and from the outside environment what will happen? These numerous sodium ions will get influxed, will come inside. And previously we have studied that influx of sodium ions contain depolarization, action potential is generated, finally nerve impulse is generated, and it gets transmitted. Here we have the dendron. It will carry the impulse from the cyton. From the cyton it will go to the axon of the next neuron. Okay, this way the nerve impulse is transmitted from one neuron to another neuron. Mind you, you must have noticed that the nerve impulse which was created in the previous neuron, that same impulse is not getting transmitted here. Okay, what is happening is here another new impulse is getting generated, but this is dependent on the previous impulse. How? Using chemicals. So we also call this as a chemical synapse. We also call this as a chemical synapse. Why we are calling this as a chemical synapse? Because this is carried out by using chemicals known as neurotransmitters. Also, just for information, you must also know that there are electrical synapses also. Electrical synapses also. Okay, these electrical synapses, what are the difference between electrical synapses and chemical synapses? No, chemical synapses, the nerve impulse is transmitted using chemicals, and electrical synapses, the nerve impulse will be transmitted using the electrical impulse will itself be transmitted. Okay, there are no neurotransmitters involved here, but these are very rare and it is out of the scope of our syllabus, so we do not need to study about this. Just keep it in mind that we also have electrical synapses. So that's all for the first part of the video. If you like the video, please hit the like button, share it with among your friends, and please subscribe to my channel. And I'll give the link to for the next part of the video in my description. Thank you for watching.